Hello everyone, I'm Koyan and today we are going to learn how to actually answer questions related to Form 5 Chapter 12. Yeah, I finally entered Form 5 1. So Chapter 11 and Chapter 12, they actually kind of intertwine with each other, overlap with each other, where some part you actually learn it in Chapter 11 and they also repeating it in Chapter 12. So before I would like to enter the question, I would like to show you what are the difference between the mutation whereby students always get confused. Let's take a look at what we actually kind of learn in chapter 4, which is not really frequently asked about how DNA can condense until become a chromosome shape. So a lot of students don't know how to differentiate gene mutation and chromosomal mutation. Gene mutation is actually whereby it happens in a specific segment of the DNA which codes for a specific protein. And then this DNA will further, will wrap around the histone protein and form a nucleosome, and then it further coil up and then form a chromatin, whereby we learned before in the prophase. Yeah, is where chromatin will condense into a chromosome. So actually chromosome they looks like that. Yeah. It's just that all the chromatin they condense together in very dense and thick manner. So they are actually chromatin right here. Okay. So yeah, it's basically a chromosome shape right now. And most of the situation, our cell exists in, our cell, right? The nucleus is normally in, the DNA lah. It's most of situation in form of chromatin. It's not in form of chromosome because it will only form as a chromosome when they are going to divide because they want to divide in a more organized manner, then they will only condense. But most of the situation, it will be in a chromatin manner because it will be more easier for the transcription and translation and yeah, protein synthesis process because of this noodle strand shape kind of thing, yeah, it's more easier to, transcribe into mRNA for the coding of the protein. Yeah. So a lot of students don't know how to actually understand. Okay, when there's a gene mutation, what it will actually affect. And when there is a chromosomal mutation, what will actually affect. So okay, let's say gene mutation. Okay, one of the base gone wrong. Okay, how it gone wrong? Maybe base substitution, base insertion, or base deletion lah. But what I'm trying to emphasize is in a normal situation, okay, there will be a complementary base pair. Then this DNA actually inside this cell, our cell. But this DNA actually can't escape the nuclear pore of the cell. Nucleus, sorry. The DNA can't actually escape the cell nucleus. So they need someone to help to transfer this code. So who is the one that helping it is the messenger RNA. That's why it's called messenger RNA. This messenger, right, it can uh, exit the nuclear pore. So when, yeah, the mRNA will be formed inside the nucleus is the one that help to transfer the code for the protein synthesis. So DNA, it will be transcribed into mRNA. mRNA is single-stranded mRNA. And then this mRNA can exit the nucleus. That's why you need this mRNA friend. It helps to transfer this message. So yeah, it transcribes into mRNA. So this mRNA will exit the nucleus and then it will reach to where? It will go to a site of protein of synthesis, which is the ribosome. Ribosome is attached to the rough endoplasmic reticulum, right? So yeah, it's whereby the translation process occur. So what actually happens in the translation process is whereby amino acid will be formed by look. Normally we will look at the codon, which is a triplet code at here, and then it will form a specific amino acid. So this amino acid, it will join in a chain. Many amino acid join in a chain together. Okay, translation process, and then it will form one codon, it will form one amino acid. And then because there are so many codons, so yeah, many amino acid form, and then they join together to form this whole thing called a polypeptide. And then this polypeptide, it has to be modified, to be packaged, modified, and folded so that it has its own different structure, unique structure for the function. So it will be modified in the Golgi apparatus. Yeah, I'm so lazy to write the whole thing. Apparatus. To be modified in Golgi apparatus to fold the protein to have its own structure. And when it has its own structure, the structure will dictate on how it functions. So yeah, this is how it basically works. And whereby when you want to say that, oh, this is not the correct way, but I'm just simply putting it in an example. So if one of the base changes, right, okay, let's say, then another base also change. Then this base in the mRNA also change, right? So the code for a specific amino acid, let's say initially you want GUA, yeah, to code for, okay, this one is a wrong example lah, but yeah, let's say it's glutamic acid. But now, because of the changes on one specific base, end up is GTA maybe, and then end up code for another type of amino acid. Yeah, sorry, I mentioned a not correct example lah, but I'm just saying what if one of the base change? And when there's a changes in the specific base, there is a different code. Now it codes for what? A different amino acid. So yeah, different amino acid and then form the polypeptide. So the folding will be slightly different, and this will eventually lead to the structure will be changed. When there's a change in structure, they may be not functional protein anymore. Because let's say this part, okay, let's say this is an enzyme. Normally it shapes in this way because active site has to be specific. What if maybe one of the amino acid right here is, the the codon, it changes, which codes for a different type of amino acid. So to be more precise lah, because of the chemical, the interaction lah, the R group interaction, but you don't actually learn this lah. So it will actually form a different folding pattern. Okay, when there is a different folding, different interaction between the amino acid, maybe it will end up changing. It will end up changing the structure of it lah. So when it end up changing structure, yeah, it will actually affect on the function of it. So maybe now the enzyme can't really function well because the structure of the active site has been changed. The structure of the enzyme will be changed, especially the active site. That's the reason why one little base, maybe you might see not much problem. It will eventually lead to quite huge problem actually. So one of the most common situation where we learn in SPM is actually the sickle cell anemia because of base substitution that occur whereby thymine is replaced by adenine. Therefore, the hemoglobin, which is the protein, hemoglobin synthesize, it tends to be, it will lead to red blood cell. Initially, it's biconcave red blood cell, but end up become a crescent shaped red blood cell. So it can't perform its function that well. And what about mutation in chromosome? So let's say there's a deletion in a chromosome. Okay, when you delete out this part, it will lead to what? A huge loss of genes. Very a lot of loss of genes. This one is just a small part of the gene. But for this chromosomal mutation is more severe because it leads to a huge loss of genes. Because you can see when you zoom in, yeah, that's the effect. So now let's take a look at this. Gene mutation consists of base substitution, base insertion, and base deletion. And chromosomal mutation, they have deletion, duplication, inversion, and the translocation. So this one you can actually refer to textbook to have a more better and clearer picture lah. But normally we would mention on a case basis is whereby sickle cell anemia is whereby base substitution occur, and deletion of chromosome is actually for the chromosome 5 whereby Cry-Duchet syndrome will occur. And chromosomal mutation is not only changes in the structure, okay, it can also be due to changes in number of chromosomes. So these four are actually leading to the changes in structure of chromosome. Can you see the structure different already? But what about changes in number? That's where we learn this part called, yeah, whereby the individual maybe have extra one chromosome or lack of one chromosome. So disease caused by chromosomal mutation. Okay, for this, it can be due to number of chromosome or structure. The structure one we learn is only Cry-Duchet syndrome, whereby partial deletion or short of chromosome 5, and yeah, results in a huge loss of genes. And then the other the other four of them is basically changes in number of chromosomes. So for Down syndrome, right, their gamete pula, their sex chromosome is actually no problem. Okay, it works fine. It's just that their autosome right here is extra one because in normal scenario it will be 44 plus XY or 44 plus XX, right? This one is happen to the female. Female doesn't have any Y chromosome. Okay, so it has an extra chromosome at 21, chromosome 21 lah. So they got three chromosome 21. That's why it's called trisomy 21. And you have to learn some of the symptoms right here. And then, yeah, tend to be slanted eyes, flat nose, protruding tongue. Okay, what about these three? I understand a lot of students don't really know how to differentiate between them, always forget them easily. All right, so if we take a look, okay, these three, the common thing is that they have a normal sex chromosome. Okay, so for Klinefelter syndrome, yes, it got extra one chromosome, but the extra one chromosome is actually occurring at the sex chromosome. It has extra one X chromosome. So this person is actually a male. But this male, he got extra X or initially male supposed to be 44 plus XXY. 44 plus XY. But now he got one more extra X. So what happens if you got one more extra X? This is how I actually remember lah. When you have one more extra X, if you are male, you got XXY, extra X. It means that you become more female, more girlish like that. Why? Because the symptoms you can see right here, the voice and chest is very similar to women. Okay, initially you got XY for men, but now you add on another extra X chromosome and not becoming more girl. So yeah, voice and chest similar to women, long legs and hands, and their secondary male characteristic is not well developed, sterile men because their testes fails to produce sperm. And what about Jacob syndrome? So Jacob syndrome is also a guy, but now he got what? He got extra Y chromosome. Initially, a man supposed to be at 44 plus XY. But now you add on one more Y. So what is the symptoms? You become more guy, not more guy lah. But basically, he tends to be taller than normal, has pimples, a lot of pimples on his face, and weak muscles, slow in acquiring speech, and learning disability. What about Turner syndrome? Okay, initially female supposed to have 44 plus XX, but now lack of one X chromosome. So yeah, she only has 45 chromosome. Therefore, when you become less girlish means you lack of secondary female characteristic, and is a sterile female. Okay, and web like neck and low IQ. You must at least know like two of them now. So yeah, this is actually how you differentiate between them. Klinefelter is they got XXY. So extra X, the guy becomes more girlish and not well developed secondary male characteristic. What about this Jacob syndrome? Supposedly, guy supposed to have XY, but he got extra Y, so he tends to be taller, has pimples on his face, and Turner syndrome because it lacks of one X chromosome, so it is not a very developed secondary female characteristic. Okay, by taking a look at this question, it is super direct and they very kind, they straight out provide to you. It actually happens a chromosomal mutation at chromosome 5. So, um, if there's a case whereby they didn't point out to you where is the problem, you can actually see it through, you focus on chromosome 5, chromosome 21, and the sex chromosome. Normally, it will occur at here because chromosome 5 is where partial deletion will occur for pride to check. Chromosome 21, if you get to see one extra, or 3 is trisomy 21, which is a Down syndrome. But if you realize these two correct, nothing wrong, maybe it's because of this sex chromosome gone wrong. Why it gone wrong? Maybe got extra one or lack of one. So if it has XXY or this one has XYY or X nothing. Okay, XXY is whereby you have extra X. So when you got extra X, this one is a Klinefelter. And the syndrome is more ghoulish. So sorry for to say that. And XYY, you become more guy. So yeah, you tend to be taller and then more pimple. Then this one, it lack of one X. So this female don't have a developed female characteristic. While Klinefelter is don't have a developed male sexual secondary sexual characteristic. So name the disease shown since this one is a partial deletion in short arm chromosome 5. This is actually a Cry-Duchet syndrome. So what are the symptoms that is faced by this person is that the when the person cry, right, it actually the infant lah, the baby cry, it really sound like a meowing of the kitten, and it can also be mental and physical abnormalities lah. So what is the type of mutation? The type of mutation we actually got learned is chromosomal mutation and the gene mutation. So this one they focus on the chromosome, right, where there's a changes in chromosome structure. So this is a chromosomal mutation. So this is actually a very direct question as long you know what is this Cry-Duchet syndrome. And this question is talking about explanation on of long-term impact of the event. What is the event? The event is whereby this yeah, US military, they will actually release a lot of herbicide, which is a chemical poison, Agent Orange, during the Vietnam War. So yeah, it caused gene abnormalities in human, animals, and plants because they are exposed excessively for a long period of time. So what are actually the long-term effects? Okay, they ask on human and plants. So since they want you to mention both, you must mention both. You can't just mention explain on human only lah. It is highly encouraged to write both if you know how to explain. So for human, there are there will be a mutation will occur towards them. So yeah, it may causes gene mutation whereby base substitution occur and increase the cancer rate of human. Meanwhile, what about the plant is that, yeah, it will lead to a stunted plant growth and the plants are actually not really safe to consume lah. Yeah. But most importantly, you have to know that it will lead to mutation. So it can be base substitution, base insertion, or base deletion. But if you want to explain in another manner, because this is actually provided by the answer scheme lah. For me, I prefer to explain it in this way. Causes gene controlling cycle to be disrupted. So actually, right, if we learn G1S, G2 phase, there are actually the DNA checkpoints. So they will actually control the there's a gene that will control the cell cycle. They will actually check if there's any issue towards the cell, the DNA of the cell before the cell divides lah, whether it's functioning well. If there's some error right there, they will actually repair the cell of it. If they can't repair, the cell will eventually undergo apoptosis. The cell will just really die on its own, like suicide on its own. But basically, there will be a gene that control the cell cycle. But because you exposed to all these mutation, right, it will cause the gene controlling the cell cycle to be disrupted. So the cell initially, it will be controlled, but now it's not controlled already. So yeah, they will just divide non-stop because supposedly cells supposed to divide under, and when they have a cell signaling, they know when to divide and when to stop. But right now, because of the gene controlling cell cycle disrupted, it will actually divide non-stop lah, divide crazily. So when there's an uncontrollable cell division, it will lead to formation of tumor. So normally in SPM, we would mention it's a malignant tumor. Actually, tumor got divided into two, benign tumor and malignant tumor. But normally we would talk about this malignant tumor whereby it is a cancerous tumor, so it's a cancer. So increase the rate of cancer among human lah. Increase on among in human. Yeah. So for my own explanation, I would write causes gene controlling cell cycle to be disrupted and then uncontrolled cell division occur. So yeah, which increase in the risk in cancer among human. And the plant is stunted plant growth and crop yield is not safe to consume. Now let's take a look at mutation that happens in somatic and gamete cell. Will produce variation and then they want you to differentiate between them. Okay, somatic cell is body cell excluding the gamete cell. Okay, the whole body cell excluding gamete cell. Gamete cell, yeah, gamete cell is basically its sperm, secondary oocyte. Yeah, these are the gamete cell. So what happens when there is a mutation in the somatic cell? So let's take a look at this question right here. Okay, it's also the same thing they want you to differentiate. That's why I show it to you. So as you can see this part right here, affected area for this person individual is the whole thing being affected, and it happens right during the fertilization. So there is a mutation in the gamete cell. And then, bless me. What about this? This happens right after fertilization. After fertilization, then the cell got mutation. Okay, somatic cell got mutation, and the affected one is only a part of it. Yeah. So the differences between them is that is straight out a mutation in the gamete. That's why it will be inherited to the offspring. For this one, gamete cell works well. There's no mutation. It's just that this offspring itself, its own self lah, has mutation. So this one is most probably involving mutation in somatic cell. So this person, right, they won't actually inherit it to the next generation. Okay, because it is not involving in the gamete cell. So the most easiest example to differentiate is like the tumor growth in your skin cell. So let's say that this skin cell, okay, you already born as a normal individual, just that one day suddenly you got tumor. Okay, so tumor in your skin cell because of mutation, yeah, it's because of your own somatic cell mutated. But will it actually inherit to your next generation, whereby your next generation will get skin cancer also? No, right? Because it only happens to you. It won't happen to your next generation. But if the gamete, okay, the gamete you produce got mutation, yeah, the gamete is involved in the formation of diploid, right? That's the reason why it can be inherited to your offspring. Your offspring will be affected, right? So that's the difference between that. So the type of cell involved, one of it is germ cell, another one is somatic cell. Ability to inherit can be inherited from parent to child, another one it cannot be inherited. So I suggest you to write it in this manner lah. Try not to write like yes, no. Okay, I know students sometimes you just write yes, no, but please don't write it so short lah. Okay, biology they prefer you to write it in a full sentence. So for this one is also quite similar. Yeah, the comparison is actually derived from textbook, so I just straight out paste it at here. All right. Now let's take a look at the next question. This one is about continuous and discontinuous variation. State the type of variation P and Q. So in order to see whether is it continuous or discontinuous, just straight out see whether can you measure it or not. Can you measure it? Yeah, you can see that their stomach size very big. It's not about measuring stomach size, it's about their mass or maybe height lah, okay? Can you actually measure it? Yes, you can measure it. What about Q? You can't measure it. Can you measure like, oh, 5.5 pattern? No, right? You can't measure it. So Q is discontinuous, P is a continuous variation. And state the two differences between variation P and Q. Yeah, there are actually a lot of differences between variation P and Q. You can state that variation P has presence of intermediate characteristic. So what is intermediate characteristic mean? It means that it has something in between them. Okay, let's take an example lah. Okay, 55 kg, 56 kg. Okay, is there anything in between them? Yes, there is a presence like 55.1, 55.2, got presence of intermediate, maybe 55.25, right? So there is a presence of intermediate characteristic. You take a look at fingerprint pattern, got intermediate characteristic? No, right? So when you're comparing, remember to write this keyword while or whereas. Variation Q has blah blah blah. Do not have, does not have intermediate characteristic. And another thing you can also mention is that variation P is influenced by, okay, your height, your weight. Can it be influenced by environment? Yes, your eating habits, your lifestyle. So it can be influenced by environmental factor. Meanwhile, for variation Q is not influenced by environmental factor. It is only influenced by genetic factor only. Okay, the continuous variation not only environmental factor lah, it can also be due to genetic factor. Let's say your parents are tall, and some more your environmental factor, you eat a lot of uh, a good diet lah, okay, balanced diet. So yeah, you're not become taller. So yeah, influenced by both. But for this continuous, let's say blood group, okay, can it be influenced by environmental factor? No. Like among these, I mean, straight out throughout these years, your blood group O, then you straight out will be blood group O. You won't change your blood group one, right? So it just influenced by genetic factor only. Won't be caused by environmental factor, then you change. No, right? Okay. Followed by the next one. I mean, there are more of them now, so you can actually refer to it. And this one is talking about reason that causes the difference in between them. So both of them, right? You can see one is taller, one is shorter. So yeah, basically talking about continuous variation. Both of them are identical twins. So their genetic composition, or you can say their genetic composition are very similar, super similar since they are identical twins, right? Okay, but after 10 years, one of it end up become so tall, another one is so short. So it is actually due to what? It's not due to genetic factor anymore. It's due to environmental factor. So you have to mention it is due to environmental factor because of maybe lifestyle and eating habit, which are eating habits and lifestyle. Yeah, that is the one difference that causes the one reason lah. One reason that causes the difference. And what about this one talking about albino person? Okay, suggest steps that taken by individual with albinism if they want to carry out outdoor activities. Okay, so in normal situation we have adequate amount of melanin. But if we kept exposing ourselves to sunlight, whereby the UV ray will actually destroy our melanin pigment, we will actually produce more melanin. More melanin is in charge of the color, okay, darker color. So when you produce more melanin, you tend to be darker in color, no? So albinism, they all produce super less melanin, okay, very less, or maybe even no. So these albino people, they are super white, yeah, very white. Okay, so albinism is a genetic disorder and then very less amount of melanin. So yeah, it can't able to filter the absorbed UV radiation lah. So it's actually, although they are super white, very pretty, very handsome, but they actually have to carry out a lot of steps when they want to go out for outdoor activities because you surely exposed to sunlight. So because they are very less melanin pigment, they tend to have a poorer vision because our retina actually got melanin lah. So since very less, they tend to have poorer vision. And when they want to go outside, it's better for them to wear sunglasses. And you try to relate to yourself, okay? You don't want to expose you too much of the sunlight. What will you do? You actually apply sunscreen, maybe wear long sleeve shirt, right? So yeah, these are actually the steps you can actually suggest. You don't have to think until so high standards. So the answers right here is just basically like that. Wear sunglasses, use sunscreen, and wear clothes that cover the skin. Okay, moving on to the next one right here, meiosis process. So X is actually can you see they are pair up, okay, pair up via synapses, and then they cross over at the chiasma site, so crossing over between non-sister chromatid, and therefore will lead to new genetic recombination. So X is a homologous chromosome. Okay, what is the effect of the process occurring in area Y? So what is the effect of this crossing over that occurs at the chiasma, which is the site, the place? So you basically would exchange the genetic material between the non-sister chromatid. So it will lead to what? Once you exchange, you actually form a new combination of gene. Then the gamete will have, because this is meiosis, it involves in the production of gamete like gametogenesis. So yeah, you have a different genetic composition. Therefore, most importantly, which is the causes of variation, crossing over is the causes of variation. So this crossing over, what is the effect? Leads to variation. So this actually more than 2 points lah. So yeah, basically you get like that. Okay, you just mentioned two of the points we'll do. The reason why it produced new combination of gene, let's say this is about the seed. Okay, yellow ringed seed. And then this one supposedly is white and smooth seed. Okay, but now you cross over, there will be exchange of DNA segment. So maybe it will lead up to yellow smooth seed, then white ringed seed. So it actually produces what? New combination of the gene. Yeah. Okay. And then this one is talking about Chernobyl accident. So a nuclear accident. Fukushima also occurred before, and now this one, this one is Chernobyl is actually very severe. So it's caused by management negligence that will release 5% of reactor. So explain the effect of this nuclear pollution on the cell cycle on the affected population. So they want you to emphasize effect of this nuclear pollution on cell cycle. So imagine what happens to the cell cycle. The cell cycle will be disrupted, will be because you can't write affected lah. Because affect is like how it affect. It can be disrupt, okay, slow down, make it faster. So you have to write it as disrupted with better word like cell cycle is disrupted. So yeah, maybe the DNA checkpoint, they didn't check it well. There's a mutation in a gene controlling the cell cycle. Therefore, the cell, they are not under this signaling when to stop dividing. So they will just divide non-stop. Uncontrolled cell division occur. Uncontrolled cell division occur, further elaboration, it leads to a tumor, formation of tumor, which is cancer, malignant tumor lah, to be precise, because cancer can be spread throughout your body. But for benign tumor, it only localized in one specific area. Let's say if you have pancreatic cancer, it only localized at pancreas, it won't spread. But if it's a pancreatic cancer, pancreatic tumor, so if you have malignant tumor in your pancreas, which is a cancerous cell, it will actually spread through your blood circulatory system or spread through the lymphatic system to go to maybe your liver, yeah, other organs, and your other organs will also get affected, will also form tumor. That's why it's divided to stage 1, stage 2, stage 3, and stage 4 for cancer. And the third one, the last one is actually explaining why lah. It's because the radioactive reactor core is a carcinogen which causes mutation. Alright, moving on to next question right here. Yeah, it's the same thing as what I mentioned just now previously. Hey, this one is actually more of factor demand, but I just put it at here. I also don't know why, but yeah, this is talking about muscular dystrophy, whereby it's a loss of muscle with mass, very weak muscle. So how are you going to use the method to treat this person? Okay, you can see this virus right here. They insert a normal virus inside, and then they inject it to the patient. So this is actually called a gene therapy, whereby they will replace the abnormal virus, abnormal gene, to a normal gene using normal gene in this virus. So it's a gene therapy. So how are you going to explain your answer? Okay. First of all, they will actually identify the abnormal gene that involved in the production of muscle. Okay. Later on, they will basically insert this. Okay, this is the virus. Very ugly, this virus. Okay, so this is the virus. Because they want to transport this gene. How are they going to transport it? They use a virus called adenovirus. So this virus consists of a normal gene, which is going to replace this normal gene now. Okay, this one is in a cell, the nucleus. So this virus will actually fills into the cell surface membrane. And this virus, right, not only they have this ability to go towards a cell, and they will do what? They will actually inject the genetic material, insert the genetic material to replace this abnormal gene with the normal gene. So yeah, now it has a normal gene, then the production of muscle, the muscle will be high quality. No, and the muscle will work more fine. So how are you going to explain? First of all, you identify the cell with a normal gene. Then normal gene that codes for production of the protein or muscle, it will be inserted to virus because the virus is the one that going to carry the normal gene to the nucleus of the cell. So virus is genetically modified to be inserted into a normal cell to replace the abnormal gene. Alright, so yeah, this one is more of chapter 13 now, honestly. Okay, moving on to this karyotype of the individual which suffer this genetic disease is also the same thing, partial deletion in chromosome 5. So it's a cry-to-chat syndrome. And then explain the cause is due to chromosomal mutation, yeah, partial deletion of chromosome 5, which leads to a change in the structure of chromosome number 5 and loss of genes. Okay, moving on to this part, okay, variation of eye color. So this variation, they present it in a bars, right? Bar graph. So there is no presence of like intermediate characteristic like this bell-shaped curve, right? So it is a discontinuous variation. Remember that discontinuous variation is only influenced by the genetic factor. Okay, continuous is both genetic factor and environmental factor. So this is causes by genetic factor. So how are you going to explain it in detail? Well, there are four things we actually learn. Crossing over independent assortment, uh, random fertilization, and mutation. But yeah, you can just straight out mention one of them and explain it in detail. So you can say crossing over occurs whereby exchange of DNA material between non-sister chromatid. You must emphasize it is in between a non-sister chromatid. And then you can also maybe like if this is essay question, yeah, you can elaborate more lah. You can say independent assortment of chromosome, whereby homologous chromosomes are arranged randomly on the equatorial of cell. Okay. And last but not least, maybe you can mention about random fertilization, whereby you produce diploid zygote that has new genetic combination. So maybe if you don't understand why it works this way, this is basically what happens behind it lah. So maybe you want to queue up in this lineup in arrange in this way, or maybe you want to arrange in this way. Because of the random assortment, independent assortment, you can see it will lead to a different combination. Okay, different gamete will be produced. Can see, right? And what about crossing over? So crossing over, let's say you cross over at here. Yeah, you eventually form a different genetic combination also. So this is how it actually meant lah. And for random fertilization is actually because yeah, there are a millions of sperm and then ended up only one will be successfully fertilized the secondary oocyte. So it's super random, right? Yeah. Okay. Moving on to this difference between variation and eye color and variation of body weight. Yeah. Variation of eye color is, can it be measured? No, it cannot be measured. So it is discontinuous variation, while variation of body weight is continuous variation. That's the most easiest difference you can state out. And followed by this question is an identical twin. Yeah, also quite similar like just now. But now let's take a look at them 20 years later. Wow. Okay, one of them become very obese. And one of them, if you focus lah, initially both of them are super fair. And one of them end up having a slightly darker skin. So you can actually identify what are the type of variations shown by the twin and discuss the factors. So one of it is actually due to, the both are actually are actually discontinuous. Discontinuous. Sorry, both are actually continuous. So I'm sorry for that. Okay. If you take a look at textbook, right? So sorry. If you take a textbook lah, the example of continuous variation they got right is height, weight, and skin color. Yeah. So it is a continuous variation. And how are you going to discuss the factor? Well, since both of them, their genetic factor is actually very similar, right? Since they are identical twins. So we have to emphasize more on the environmental factors. Okay, why this person end up become so dark in color is because of the light she exposed. And why this person end up so obese is because of the lifestyle and eating habit. So she tends to have a sedentary lifestyle and she tends to have an active lifestyle. Eating habits you can also emphasize, maybe she eat a high fat food. Yep. So these are the things you can actually mention right here. Continuous variation, you have to state first, then you only explain about the factors. Alright, so that's all for this chapter, and I will see you in the next video. Thank you so much for watching, and don't forget to click like and subscribe. Okay, bye bye.